Welcome, welcome everyone to the sixth event in Contact's Produce Safety webinar series. This series will tackle a different fresh produce safety topic every month through a dynamic webinar series that will bring together industry, academia, and regulatory minds to solve some of the produce industry's biggest and most timely challenges. This series is part of a larger industry outreach and risk mitigation project called Contact, or scientific challenges and cost-effective management of risks associated with implementation of produce safety regulations. This project is supported by the Specialty Crops Research Initiative from the USDA National Institute of Food and Agriculture. Any opinions, findings, conclusions, or recommendations expressed in these presentations are those of the speakers or the panelists and do not necessarily reflect the view of the U.S. Department of Agriculture. This webinar series, coupled with other research and extension activities, is part of a collaborative effort across 10 different universities or government institutions to enhance produce safety knowledge and compliance across the industry. We are so grateful to our collaborators for their efforts and support of this webinar series. You can see their logos on the screen. This webinar will continue, or this series will continue next month and feature the talented and knowledgeable Dr. Bob Whitaker and a panel of experts from industry and academia to untangle the complex and often challenging web that is the produce supply chain. This webinar will include, of course, an interactive Q&A with our audience, and it serves as the season finale to Contact's Produce Safety webinar series, and it is sure to be a special treat and dynamic end to the season. Please join us May 12th of this year. This multi-year webinar series will take a break over the summer months and pick back up in the fall with even more industry-proposed ideas, including diving deeper into agricultural water and risk-based thinking, traceability, sanitation, among others. Season 2 will not disappoint. As always, if there are topics you want to hear more about, please let us know in the comments, webinar surveys, or on our social media pages, or you can always reach out to me, Laura Strawn. While this webinar series lets us meet with you once a month, that's just not enough for us. We'd love to #make contact with you about today's webinar or other produce safety news and research on our Twitter and Instagram pages. Follow us on these platforms at PS_SCI. A quick note for today, if you would like to submit a question for our speakers during or before the Q&A session, please submit your questions through the Q&A box and not the chat box. Now, let's welcome our wonderful panel of speakers today. We have Dr. Faith Critzer, who's an associate professor at the University of Georgia in the Department of Food Science and Technology. She joined UGA in fall of 2021, and prior to that, she was an associate professor and produce safety extension specialist at Washington State University. Her research and education program is focused on identifying food safety risks tied to the production and packing of fresh fruits and vegetables, as well as identification of and education on risk mitigation strategies. The most recent work from her lab group was focused on Listeria control in the packing house, pre-harvest agricultural water treatment in order to inactivate bacterial foodborne pathogens, and validation of antimicrobials in post-harvest flumes using industry-relevant conditions. She also has a passion for communicating science-based best practices and outcomes of research to the produce industry. Blanca Ruiz Jacksuwanga is a research coordinator at the University of Georgia in the Department of Food Science and Technology. Prior to that, she was an associate in research at Washington State University working with produce safety. She also got her master's degree at Washington State University, focusing on environmental monitoring programs to control Listeria in apple packing houses. Last but not least, Dr. Alexis Hamilton is a food microbiologist working as a post-harvest associate in my lab here at Virginia Tech. She has spent the last 6 years conducting research on environmental monitoring programs in fresh produce packing environments, Currently, she is also working collaboratively on experiments evaluating the survival and transfer of foodborne pathogens on fresh produce and packing materials, describing food safety indicators in soil, and assessing food safety attitudes and management practices of growers, farmers, and other stakeholders to coordinate and optimize outreach materials. So, welcome all, and Faith, the floor is yours.
Thanks so much, Laura, and I will get my screen up and going. Wonderful. Okay. So, as Laura mentioned, I'm going to be kicking us off today. I will say that I'm trying to cover a lot of area in 20 minutes. So, hopefully, this is going to be recorded. So, if you're brand new to environmental monitoring, I will say you're going to pick up on a few things, but go back and re-listen, because every time you listen, you're going to, and as you get more knowledge, you're going to definitely, more things will resonate with you. So we've delivered this talk to some grower meetings over the past year. Clearly, there's a lot of work that's been taking place, for decades in this area. I will say that, it's been a team effort as far as developing the slide deck with all these individuals shown here, including Laura. And we'd also like to thank Dr. Michelle Danila, as everyone knows, who works in Extension. We're all about borrowing each other's pictures and slides and things along those lines. So really appreciate that input. So I'm going to be starting us off with thinking about how do we actually go about developing an effective environmental monitoring program. What we tend to see is when people are in the mindset of checking a box, do they have an environmental monitoring program? A lot of times they're not making the best use of those resources. So giving you some food for thought, I will say that all of you that are in the produce industry, you are experts at some level. And we're going to give you a few things to think about today, but you know your own commodity, you know your process really well. So you're going to have to think about, within the context of that knowledge, what you're hearing today. And are there things you want to dig deeper into? Are there some things you may think about changing if you're at square one in trying to implement an environmental monitoring program? I would say that there's a lot of different ways you can go. And we're going to provide a little bit, very tip of the iceberg information to get you started. And then Alexis is going to follow up with looking at what is some of the recent findings from literature say. We've had a lot of efforts going on all throughout the United States, looking at packing houses is a unique situation. And fresh produce is a unique situation, being a raw commodity as well as ready to eat. And then Blanca is going to wrap us up with looking at, we know that, hygienic design when it comes to a lot of our equipment is just not exactly, we're not the dairy industry and things along those lines. And we're still going to, be utilizing this equipment for a lot of quality purposes, right? So how do we go about actually taking care of sanitation issues as they arise? So when we're thinking about environmental monitoring systems, really what we try and use these systems for is an early warning system. And we want to develop really robust measures so that we can actually prevent a loss of control from occurring. Not responding to your environmental monitoring program is really a lot like your house alarm going off and you letting the burglars break in and take everything without calling the police, right? So we want to think about these systems in that way. We really want this hopefully to describe the processes and the activities that need to take place to characterize and monitor the quality of the environment. Typically, when we've gone back and seen high profile outbreaks where there was linkage to packing house contamination, it was pretty widespread contamination. If they had, a better environmental monitoring program targeting some of these foodborne pathogens, then they probably would have picked up on it earlier and been able to do something about it before it got to the level it did to cause such extensive cross-contamination. We're going to look at evaluating the effectiveness of the hygienic practices. So a lot of that is around the sanitation program. And then also looking at so that we prevent possible microbial contamination of food products. This is also going to help you, if organized correctly, really identify what are your weak points when it comes to issues with either indicator organisms or foodborne pathogens specifically? And what areas do you need to focus on far more intentionally within your program? I think one of the big misconceptions when people are first getting started is that they look at finding positives, or if we're talking about indicator organisms linked to sanitation effectiveness, so something like an aerobic plate count or total coliforms, where we're really quantifying, or even ATP. They think about when they get, positives or exceeds limits as a failure. And I would say, no, it's not a failure, because ultimately your Sentinel program's working. It's telling you when things need to be checked in on, and so you can resolve issues. You should get in the mindset that you're going to find positives. You're going to find these organisms. Given that produce is a raw commodity, it's going to come in with hopefully very low levels of contamination on it. What we want to do through our environmental monitoring program is really wipe out our transient microorganisms that are going to be coming in and out, make sure that they're not setting up shop and giving us greater issues through that. So we're really trying to get in that mindset. We're going to have positives. We need to have ways to respond to our positives, and we need to always execute effectively and make sure we're fully planning our brunt force behind it so it doesn't become a larger issue. So make sure you're preparing yourself as well as your team for this reality, and make sure that they know it's not a failure and that they're really doing their job well, and you're getting better and you're learning more about your systems as a part of that approach. I will also say that an EMP really does build upon a lot of your already existing programs. And you need to make sure these programs are really well buttoned up before you go and start throwing a lot of funds at an environmental monitoring program. Really, we want to, of course, look at everything's the basis of good agricultural practices and good manufacturing practices, of course. As part of that, sanitation is key to environmental monitoring so many times, and controls that we have in place from that sanitation program. If you don't have this buttoned up, the environmental monitoring program is just going to be able to document that you're not doing this job effectively. So first, make sure these things are happening the way you think that they're happening. We can also look at sanitary design and where, again, we may have some weak points within our system. Zoning controls, a lot of times in produce, we're working in one big open facility, but if you look at more manufactured foods, and there are some produce commodities which have this, where you do have, essentially, we're trying to get the produce cleaner and cleaner as it goes through our system, where you do have differentiation as far as physical space and things along those lines, trying to keep essentially our finished product from commingling with our raw product. Employee hygiene training, a lot of times, it is also key when it comes down to things being executed sufficiently, as well as preventive maintenance when it comes to a lot of our equipment. When people ask me what needs to be in their environmental monitoring program, this is the question that I turn around and ask them. Why are you developing an environmental monitoring program? Because your approach for actually getting your program set up is going to be slightly different depending upon how you answer. And you could answer yes to many of these different categories. So the first is risk management. I'm trying to prevent, a foodborne pathogen from cross-contaminating my product as I'm packing it out. Many times those programs are going to be focused around either indicators closely linked to pathogens on food contact surfaces, and sometimes even looking for the food pathogens themselves away from those food contact surfaces. We also look at cleaning and sanitation verification as another approach that a lot of people are implementing an environmental monitoring program for. You go through the action of cleaning and sanitizing a surface, but how well did you do it, right? So by actually looking at the remaining microorganisms that are present, a lot of times will involve ATP here, which is not linked to microbial level, but again, a really good indication of is there organic matter present, so that you can get a really rapid feedback as to how well you're doing in that realm. A lot of times it may be regulatory requirements. Most of you are going to be under the produce safety rule, but for those who may be under the preventive controls rule, again, since we are dealing with a ready-to-eat commodity, there are requirements there around environmental monitoring. Auditor and buyer requirements is another one that we hear about, as well as, investigations and really trying to figure out for cause, what's going on within a system. So when we think about an environmental monitoring program, it's going to build upon your knowledge and your team's knowledge that is tasked with developing the program, a basic understanding of microorganisms, why you're targeting what you're targeting, and explicitly what that means as far as what answers you think you're going to get out of it. Unfortunately, in the 20 minutes we have today, we're not going to get all the way there, but that's what a lot of the resources that we'll link to after this webinar are going to help supplement your knowledge if you haven't gained that before. Also, there's a lot of people who develop entire, multi-day workshops on this topic. And Alexis is also going to share a little bit of information around some fun things we have coming up as part of this project. Again, what's the reason that you're performing the environmental monitoring, the purpose of your program? How's the data going to be used? Again, it's very specific for your commodity as well as your packing house. So make sure you have everybody on board from management all the way down to the people implementing the program, because that's the only way you're going to make it effective. Also, know, again, you're going to get positives. You want to find them. We expect them to be there. We want them to make sure that they're cleared and they're just transient positives. But again, you have to determine this, and we'll talk a little bit more about that at the end of this presentation. So we see differences in micro populations based upon their ability to thrive. Listeria monocytogenes and Salmonella are two pathogens a lot of times we focus with environmental monitoring. That's because they've been linked to cross-contamination within the packing environment. We'll talk about that a little bit more in Alexis's presentation. We're also going to see that, different facilities based upon historical outbreaks and things along those lines are going to worry about different microorganisms. So sometimes we have a strong linkage to Salmonella, sometimes we have a strong linkage to Listeria, sometimes it's both. And so your approach is going to differ slightly in what organisms you target within your EMP. Microorganisms can always find ways to survive in the environment. They are very much thrifty and survival of the fittest. Talk a little bit about biofilms in our next slide, but ultimately it's really trying to figure out how can we prevent biofilm formation as well as sanitizer resistance. So within biofilms, again, biofilms are not just a monoculture. They're a mixed species culture of a lot of different microorganisms. The worst case scenario is when you get a biofilm forming that has a foodborne pathogen present. Why? Well, as we can see in this nice picture down here from Valentina Trinetta, we can get basically individual cells growing up into very dense populations that are not just bacterial cells. We also see that exopolymetric substances basically that will protect them from sanitizers that are going to be exposed. And essentially, once they're at such high populations, they will break off, be protected from those sanitizers down below, and then they will cross-contaminate crop as it's packed out. Know that a lot, we have a perfect example here, a piece of equipment that's really great for entrapping organic debris and other things. Really hard to get out considering the shrouding and everything. Its design is really common in our packing houses. So how do we make sure that we're not having a biofilm form on this, folks? Again, our environmental monitoring program is going to be our sentinel there. So a lot of times when we're trying to determine exactly where am I going to sample, well, ultimately what we're trying to have you think about is when does a hazard, so a hazard specifically being the foodborne pathogen that you're trying to target many times, or an ineffective sanitation program. And when is that going to actually become a risk for the product I'm packing out? So think about your layout, right? And this is going to differ for everyone. And then think about how your product moves throughout the packing house, all the places it goes and all the surfaces it touches. Think through historical outbreak data. And again, that's going to be as well as recall data. If you have any environmental monitoring results already in hand, that's going to be key for you. If not, there's going to be a little bit of investigation which will take place up front, as well as our published literature. So then we think through, well, what's actually the likelihood that these hazards are going to contribute to contamination of my produce? So we think about proximity. So we tend to think about our zone ones or our food contact surfaces being the highest risk factors, and then essentially radiating out from there. And as we get farther and farther away, we should see less of as an actual reality of cross contaminating that crop. And then ultimately, how well to actually execute on cleaning and sanitization. Sometimes we can have great crews that are well trained, and without these EMP programs, we just don't know that we're not hitting it with enough when it comes to cleaning a piece of equipment, and we need to give more effort there, or reevaluate how we're going about cleaning and sanitizing. Ultimately, we're trying to look at identifying harborage niches when we think about actually, okay, I think these are my biggest issues, and trying to manage those risks and determine when they do become an issue. A lot of times we're thinking about hygienic design. We have a few examples coming up. Ultimately, what we're trying to prevent is cross contamination. And we're really trying to prevent, both not only the things that are direct food contact surfaces, but those things that move all throughout our packing house. And by things, I also mean people as well, forklifts and things along those lines that are commonly moving commodities. We're also going to try and establish acceptance and rejection criteria so when, and your team knows clearly when we need to have corrective actions. So when we think about our biggest risk factors, here is a plethora of different things that will cause us problems. So a lot of times we have aging infrastructure, and we have basically areas that stay moist, even though they're not direct food contact areas, as is shown in this picture, giving us issues. Drains are always going to come up as a big risk factor, especially when we think about Listeria controls. So how do we go about managing drains and as a weak point within our packing house? This is on a floor, but again, this is a great hollow area, and then joined onto the floor, all sorts of great little niche points for bacteria to come in here. And ultimately, this is a forklift stop. So again, we have a lot of product potentially exposed here just below. Again, preventive maintenance within our belts and things along those lines. Cold rooms is shown here. Improper welds, or unsanitary welds that essentially allow for greater surface area and niche points for bacteria, as well as modifications to equipment that shown here in these two pictures, and then pooling water. So all of these things are things we regularly see within the produce packing house, and essentially we need to try and manage effectively and have our warning systems out. If we're thinking about our EMP as a cleaning and sanitation verification strategy, um, ultimately what we're trying to do is verify that you're following the procedures that you developed. They're actually working robustly, and they don't require modification. Without this, you're not going to know. And ultimately, a lot of times I say it also comes into looking at, am I doing what I said I'd do in my sanitation standard operating procedures? When we think about monitoring, this is a berry harvester, showing, a modification right here down on our catch plate. Uh, this piece of rubber that was put in was done so to prevent noise. But I guarantee you these rivets, and I know from talking to this producer, these rivets and this rubber essentially entrapped everything that came into it. So once we talked through kind of risk points, they decided to take it out. But they first picked up on it being an issue because they implemented an environmental monitoring program. So in our monitoring, if you don't already have an EMP, you're going to want to establish what we call baseline data. This is where you go out and you cast a wide net looking at all of the populations of microorganisms that you're targeting within your program, and really try and evaluate what are the common populations I see, what are my biggest risk points, and essentially provide more data into what sites are you going to sample more intensely and what sites are going to be more of an issue for you. Again, it's going to give us our process control verification and be that systems based checked. And it's really going to help us solve a lot of our issues that, since microorganisms are invisible, we would not otherwise know about. When I'm going to talk a little bit about corrective actions just to anchor us into, and not steal Alexis' thunder, but to anchor us into zone one's our food contact surfaces. A lot of times we're looking for anything from ATP to actual microbial indicators like generic E. coli, coliforms, total anaerobic bacteria, ACA, or total aerobic plate counts. We don't sample out of practice pathogens on zone 1 surfaces. If we found them there, then we would have a likelihood of thinking our product had been cross-contaminated, we had backed out on. So when it is done, it tends to be for investigational type purposes. Zone 2s are just adjacent, so a lot of equipment supports. Again, we can have indicators, and in some instances, pathogen sampled there. And zone 3 is everything else within the packing house: drains, floors, walls within that area. And zone 4 is outside the packing house. When we think about sampling for microorganisms, we look at our aseptic technique as being paramount. With ATP, it's not, again, you have organic matter on your skin, but when we're sampling for microorganisms in the environment, we don't care about what's on your skin. So by using good aseptic technique, it's really imperative. We have a couple different options. We can have our swab style stylus, is shown here, for sampling smaller surface areas, as well as our typical sponge type swab. This is either cellulosic material or a synthetic material, is shown here. And essentially, the difference between these two is ultimately how large the surface we're trying to sample. We know that we get better recovery microorganisms if we have moisture present. We're going to want to use sponges or swabs that have a neutralizing buffer. DE or lethine broth are examples of these. Why a neutralizing buffer? Well, if I'm sampling after I've applied a sanitizer, I want to neutralize that sanitizer. So I'll do that through that DE or lethine broth in my swab itself. Again, we're going to go through and swab our surface. A lot of times if we're sampling for indicator organisms, we'll sample a 4x4 inch square. And when we go to larger surfaces, when we're looking for things more pathogen associated. Again, if the surface is very small, then you're swabbing a very small surface. Ultimately, when we get back data, we're going to want to do a really robust root cause analysis to try and disprove underlying cause of the event. We're going to look at data collected and through additional environmental swabs we'll follow up on next, as well as visual investigation, which does tell us a lot of the story sometimes. And that visual investigation could even be looking at your sanitation crew perform a sanitation event. We'll begin to then be able to eliminate categories and focus attention on others, and essentially with the goal of ultimately trying to improve the whodunit within our root cause analysis. This should drive changes to your programs and hopefully prevent occurrences in the future. All corrective actions that are triggered should be implemented in a timely manner and documented. It is recommended that if you get a presumptive positive result from an environmental sample, that you treat it as if it is a positive. And all of our corrective actions or measures need to be predetermined and specific to the 4 zones. A lot of what we talk about when it comes to trying to figure out what is going on when you get a positive, we will introduce this concept of vector swabbing. So on this example, if we had this brush bed, we got our positive here, we will fan out and take anywhere from 5 to 15 additional swabs from the surrounding area. This helps us determine how widespread the contamination is and will help us really hone in on exactly how we need to go about rectifying the situation. Typically, we're going to want us to troubleshoot samples and not pool them so that we can actually determine between all these 15 sites exactly where we have positives and not. These are some things to think through when it comes to your corrective actions. For our zone 1 surfaces, we start with vectoring, right? So we want to really try and span out and really see how far spread the contamination is. We're going to then want to determine if the line needs to be broken down for visual inspection and swabbing. It's especially important you think about breaking down the line once you get repeat positives, right? For cleaning and sanitizing, we will then go through and thoroughly clean and sanitize the line in the surrounding area using our dry, our wet, or our controlled wet cleaning procedures. We then go in for our 4th step, being our pre-op, where we actually go through and conduct our pre-operational inspections. And then we take additional vector swabs to make sure our sanitation event was effective. And then we continue to hit this site more frequently in our subsequent sampling so that we make sure that essentially we can clear our positive. Typically, after we get 3 subsequent negative samples, we're able to really implement and say that we've cleared our transient positive, hopefully, that we've established and we've taken care of the issue. For zones 2 and 3, we see a lot of the same approach. I would also say, since we're radiating out within our production environment, consider if you need to quarantine the area. And then, of course, we're going to want to try and figure out exactly how widespread. The likelihood as you get from zone 4 to 3 of finding a positive is going to be far greater than hopefully what we're seeing on our food contact surfaces. Again, if you're sampling for pathogens, which we can have occurring in zones 2 and 3, think about stopping production and prepare the system for a cleaning and sanitation event. Really, when we're thinking, is trying to prevent cross-contamination onto our food contact surfaces. Zone 4, we're going to have the most likelihood of finding a positive. Again, this is areas outside the packing environment where we have more pressure. Ultimately, you're going to find them there, and you want to make sure they don't proceed into the packing area. So limit access, think about quarantining, perform selected zone 3 sampling to make sure you don't have spread. And then again, try and clear the transient that you have there. So as far as some final thoughts, you're going to need to have corrective actions that are in place. We've given you a few things to think about here. Ultimately, be consistent in your approach and document it. Document resolution. We're expecting to have transient positives that pop up, but we really want to make sure that we're documenting that we did handle the situation and that we did clear them. They aren't resident. Think about your root cause analysis, that fishbone diagram, and try to improve upon, are there programs that need to be improved upon or flow or something along those lines that are giving more headaches that you can somehow rectify in your process. And be purposeful in building and implementing your environmental monitoring program and evaluate it regularly, at least annually. But I bet you're going to have more frequent evaluation. So thank you all. And I will turn it over to Alexis.
All righty. Thank you, Faith. I'm going to go ahead and at this point now, you should be looking at this slide that has purpose on the front. Everybody feel free to throw some information in the chat if that's not what you're seeing. But really what I'm guessing that you all are here today to really learn about is you're wanting to know a few things, right? You're wanting to know what sorts of organisms are we looking for when we implement an environmental monitoring program. And then you're also going to want to know where those organisms are coming from or where they're starting to show up. And then I'm guessing as well, you're going to want to know this third piece about how can we use this information to improve our own environmental monitoring programs today. So that's what we're going to talk about. All right. Before we jump right into talking about some of the research, we're going to build on the context of this environmental monitoring for food safety and packing house that Dr. Critzer just spoke about. And I'm going to talk about some of the common indicators that are used in these programs. So specifically what those are, how we might use them, and then what the literature says about how and where they tend to show up in the packing environment. Then we're going to move on and talk a little bit about Listeria. So we're going to look at what are our best indicators for this organism, where should we be looking for it, and then how can we incorporate that sort of sampling approach into your program. And then I'm going to wrap it up by reviewing some of the key takeaways that the scientific literature has given us. And I'll give you 3 things that you can do today with the information that's in this presentation to improve your own environmental monitoring in your facility. All right, so one of the things that you might start monitoring for is an indicator organism. And this is a group of organisms, basically, that can give us an understanding about what the general microbiological condition of a food or an environment might look like at any given moment. There are some examples on the slide here of some of the indicator organisms that you may be familiar with and that are commonly evaluated in the literature. And this just means that if you want to gain a better understanding of where these organisms are in facilities like yours or under conditions maybe that are similar to those in your facility, there is some existing data that we're going to talk about here in a couple slides to help you understand how and where that you might start sampling for them. But first, I want to talk a little bit more about indicator organisms. Specifically, we're going to talk about indicators of bacteria and how they might be useful in your operation. All right, so If we think about what an indicator organism can do for you or how you might want to use them in your EMP, they can be really good for helping you determine the hygiene status, so how clean or how sanitary your produce packing equipment or environment is. It can also be really useful in validating and verifying your cleaning and sanitation programs, right? So validation would be proving that your cleaning and sanitation is effective at reducing these microbial populations, and then verifying that that works in real time. And they can be really good for helping you determine if a contamination risk has occurred in your facility. So depending on the type of indicator organism that you might pick, some can be closely linked to the likelihood of fecal contamination in your crop. So if you're already monitoring for these, it can help you stay ahead of the curve on making decisions about food safety. And then, of course, the longer that you do this, the more data you're going to collect. And then you can use that data to help you evaluate the food safety trends that you're seeing in your facility. So this is going to include things like, are your counts higher in the packing house in one season compared to another? Do you notice a trend like this with a particular crop, or maybe once you've implemented a new piece of equipment? And then one of the coolest things that indicators can do is they can help you figure out whether or not a growth niche actually exists in your facility. And Dr. Critzer mentioned a little bit about things that might impact whether a microorganism is going to survive or grow. But what's important here is that this is an area where you have some control within your facility. Right? So monitoring for indicator organisms can help you find some of these areas where maybe plant debris is getting trapped, or maybe your equipment isn't getting cleaned very often, so that you can then adjust your practices to hopefully minimize that risk of contamination. Now, I also want to mention a few things, well, really one major thing that an indicator organism cannot reliably do. And they're typically not very good at telling you whether or not a pathogen is present at the spot that you're sampling. And I know that this is really disappointing, but the literature and the research is pretty clear that these groups are not very strongly or reliably linked together. Okay, so I want to talk a little bit now about how you might use a couple of these indicator organism options. One of the ones that you may be familiar with, because this is one of the most common ones that we see, is the aerobic plate count, or APC. And when you sample for these kinds of organisms, it's going to enumerate bacteria, so typically not yeasts or molds, that are present at that sampling location and then can grow in the presence of oxygen. But because this isn't a very selective indicator, right, it's not going to tell us about what types of bacteria are present or anything about pathogens. It's really most useful to use this indicator as a way to validate and then verify your sanitation procedures, right? So you can use this to actually count the bacteria that are on a surface through these practices of enumeration. And then you can establish kind of a baseline and use that as your way of adjusting your practices over time. And if we look at another one that's pretty common, we can think about our coliforms. This is also a group that's going to be really good to use if you want to evaluate the sanitary quality of your facility, because coliforms are actually this broad group of microorganisms that are commonly found in the environment or on soil or on vegetation. And so once you do this validation piece of your sampling protocol for coliforms, if you get an elevated result, or maybe something that's a number that's higher than what you were expecting, that kind of increase might mean that there's some sort of environmental contamination event that occurred. E. coli is also going to be a member of this group. So this indicator is often used as a way to monitor for insanitary conditions or post-process contamination as well. A less common indicator that we're seeing that seems to be getting a little bit more popular, especially in dry facilities, is this group called Enterobacteriaceae. And you could use this indicator in much the same way that you would use coliform testing, because all coliforms are Enterobacteriaceae, but not all Enterobacteriaceae are coliforms. So you can see this if you look on this picture over here on the right side of the slide. You've got your coliforms in this sort of peachy orange color. And then outside of that, that's encompassing it, is this yellow category of Enterobacteriaceae, right? But what's nice about this Enterobacteriaceae, or EB, is that this can provide some additional information about Salmonella, because Salmonella is not a coliform. So if you're operating more of a dry facility, this might be a more useful indicator for you. Okay, so now we're actually going to get into the fun part, which is talking about some of the data. So I'm going to share with you some of the results that we found and looking at some of the published literature. I want to say first that the findings that we're going to talk about over the next few slides are the results of looking at publicly available data, meaning this is data that's been published in a peer-reviewed journal, and it's not going to include any kind of in-house environmental monitoring studies. We're not really going to talk about that. We're just going to talk about what's published and publicly available. So basically what I did was I went through the scientific literature and I found 3 studies that were done in fresh produce packing houses that I thought would be really relevant for us to talk about today. And I picked them because they all had tables that look like this on the right-hand side. So you can see that they've got this first column that talks about these specific unit operations or where specifically they went through and collected some samples from. And then it's also going to include numbers. I wanted to see actual counts of some of these indicator organisms. And you can see on some of these other columns, we're including some relevant indicators that would be used in fresh produce packing houses, like the aerobic plate count, Enterobacteriaceae, coliforms, and E. coli. So 2 of them also gave a little bit of a mention of how time might impact some of these numbers. So we'll talk about that here in a second as well. But basically, I went through all of these and I noticed some of the important highlights that they all kind of shared. And that's what we're going to talk about here on the next couple slides. Right, so if we think about the kinds of trends that we're seeing in indicator organism monitoring for EMPs, the first thing that I wanted to mention is that we typically aren't seeing many of these organisms on our food contact surfaces, so those zone 1 surfaces that Dr. Critzer was talking about. And even with our APC indicator, right, which is our least selective option, they still tend to stay below 3.5 logs of bacteria. But we do start to see these numbers go up when they're in either hard to reach areas on the packing line. So this might be areas where you can't clean or sanitize very well because maybe you can't reach it or because you'd have to completely disassemble the line to be able to get to that area. But something else to think about is that because they're present in such low numbers, it can be hard in some of these areas to actually calculate and show a log reduction when you're trying to validate your cleaning and your sanitation programs. Enterobacteriaceae, or EB, and total coliform counts, or those TC, were also observed to be lower in dry areas of the packing house, except in locations where cleaning and sanitation activities were not regularly happening, right? So if you get an opportunity here for more nutrients to build up, where microorganisms maybe then might be able to use them to survive or to grow. But the literature is also showing us that these total coliform populations tend to be higher on wet conveyor belting than on some of our other equipment. We also saw that equipment that moves around a lot carries higher APC, or aerobic plate count, and coliform loads, right? So in this sort of situation, you want to think about your lugs or your bins or any other equipment that is either used over and over again before you clean and sanitize it, or that is moved in and out of the packing house a lot. And then I mentioned that 2 of these studies looked at this time component of sampling, and they found that indicator organism populations went up as the day went on, right? But an effective cleaning and sanitation program can help with that to be sure that you don't exceed your limits during production, right? So this is a bonus study that I'm going to touch on really quick. This was done in 8 stone fruit packing houses where they collected swabs from a sorter before and after cleaning to basically show that these activities were effective at reducing populations of both APC and total coliform indicators on your food contact surfaces. And so I'm sure a lot of people are looking at this and saying, well, that's not that much of a log reduction. And that's correct. That's not the point of why we clean. The reason we're cleaning is to remove some of the organic matter buildup or plant debris that might inhibit the ability of our sanitizer to be really effective at then reducing these microorganisms. So if you do a good job cleaning, that's going to make it easier for your sanitizer to actually be able to reduce the microbial loads of whatever organisms might be on those surfaces. Okay, so where our indicator organisms can tell us about growth niches for microorganisms, Listeria species can actually go one step further, and it can tell us about harborage points for the pathogen Listeria monocytogenes. This is an index organism. It's the best one that we have right now for telling us about what Listeria is up to by looking at some of its family members. If you look at this picture over here on the right-hand side, you can kind of see that we've got Listeria up top, and then broken down at the end of each of these arrows is several different species of Listeria. And you've got monocytogenes there in red. because this is really the main one that we're concerned about when we think about a foodborne pathogen. These other ones are not considered pathogenic for humans. And so when we look at Listeria species as a whole, as a group, this is a sampling group that is not considered a pathogen, right? So this is going to tell us about whether or not Listeria monocytogenes could survive or grow in that environment. Right? So that makes this indicator or this index organism of Listeria species good for a zone 1 food contact surface if you were going to test for it in a packing house. And this group is also going to tell us if you have a harborage point developing on your equipment, right? So emphasizing really whether or not the survival conditions for a pathogen are met in your facility. And then this group is also really important to include when you're sampling in a wet or a mixed packing house facility, because moisture control is really important for making sure that Listeria monocytogenes can't survive or grow in your operation. Right, so I did something very similar with these that I did with the indicator organism data. I went through the literature and I looked for anything that had been published in the last 5 years that looked at Listeria species on either food contact surfaces or on these non-food contact surfaces. And I looked for a table like this. I wanted them to tell me exactly where they were sampling from. And then I also wanted us to give them to give us a clear indication of presence-absence data. Did they find the organism? Did they not? All right, so what sorts of trends are we seeing on our food contact surfaces for Listeria? Firstly, equipment that's a part of the washing, waxing, or your drying steps tend to have Listeria detected from them much more often. Right? So specifically, we're concerned about brush or roller beds and conveyor belting. But then on the other end of that spectrum, we've got areas like the dump tank and the packaging line that didn't have as many positives. And that's typically because those are areas where either a continuous sanitizer is applied to that surface, or those surfaces tend to stay dry during the operation. Next, we're also seeing that moisture matters. Listeria was much more commonly isolated from wet surfaces than drier ones. And then the data is also showing that detection increased throughout processing. And this really isn't a new trend that we're seeing. This is actually very well established because it takes time for Listeria to work itself out of the equipment, or maybe as it might come in with the crop, right? Which is why guidance documents often recommend sampling for Listeria once you're a few hours into production. And then this last piece, this was something that these studies saw, which was that the way that you manage your packing house matters. This is not something that these papers explicitly studied, right? So they didn't go out to try to prove this piece of information. This finding, which is this idea that Listeria detection on food contact surfaces was higher in some packing houses compared to others, was something that emerged as these research teams were conducting their sampling. Right. So, but we know that there's things like consistent and effective cleaning and sanitation programs, good food safety practices, and then regular training of employees that can be really helpful for helping you put your best foot forward when you're trying to minimize detection. Now, if we think about our non-food contact surfaces, we're seeing very similar trends, right? So the areas underneath your washing, waxing, and drying operations tend to have higher detection, right? So this is going to be thinking about places like your floor or in your drain samples. And then we're also seeing that higher detection was also seen at entry points into the packing house. So you want to watch any equipment or personnel that are regularly coming and going in and out of these points. And then we're also seeing that Listeria was less commonly isolated in areas under the dump tank or the packaging line, again, where maybe there's a continuous sanitizer applied or the process tends to stay pretty dry. Once more, we're seeing that moisture matters. So higher detection was observed in wet areas compared to our dry counterparts. And then the type of movable equipment also mattered here. So it was the equipment that was either hard to clean or rarely cleaned that posed the larger problem. And then again, the way that you manage your packing house matters. These studies didn't seek out to prove this information. This was, again, just something they observed during sample collection. So I just ran through a lot pretty quick. So this is just a quick summary slide to kind of help us consolidate some of the information that we've talked about as you're thinking about enhancing your EMP. If you're looking for indicator organisms, you want to watch those brush rollers, conveyor belts, hard to reach areas, and then any equipment or materials that move around a lot. And then for Listeria species, when you're sampling food contact surfaces, pay special attention to those polishing brushes, surfaces under fans in your drying operations, or your conveyor belts. And then on the non-food contact surfaces, continue to watch those floor and drain samples and any equipment that maybe moves around a lot, like a forklift or something that's, again, either difficult to clean or isn't cleaned. And this is the part where I'm going to tell you how you can use it today. So first, I would recommend that you use the count data that was presented on the indicator organism section to help guide you in setting your own limits on different parts of your packaging line. Next, use the presence or absence data from the Listeria section to help you select your sample site. And do remember that a good environmental monitoring program is able to find Listeria when it's there. So you should have a mix of those hotspots for detection and the not so common areas as a part of your sampling plan. And then lastly, you want to compare the data that you currently have about your own operation to what was presented today, and then ask yourself, where can you minimize these factors that influence microbial growth and survival? And then how can you adjust your cleaning and sanitation program to better target those hard to reach or difficult to clean sites in your facility? Then, like Faith said, I just want to mention really quickly that as a part of this contact grant, we do have a very collaborative group that is working together to put out this online, completely asynchronous course about a foundation in environmental monitoring program. So this should be coming out here in the next couple months. So be on the lookout for that. And then I also want to say that if you have any questions or want to read any of these articles, we will be including them in that key takeaways document. And with that, I will say thank you for listening. Looking forward to the Q&A. And Blanca, you are good to go.
Thank you, Alexis. Can you see my screen? Okay. So following with our agenda, I'm going to present some sanitation research that we've been doing on packing equipment surfaces. So, but first of all, why are we focusing on these equipment surfaces? First, because previous foodborne outbreaks or recalls of fresh produce have been linked to cross-contamination with Listeria monocytogenes and packing equipment, specifically in two of the most lethal outbreaks, the 2011 cantaloupe outbreak and the caramel apple outbreak, were linked to contaminated surfaces. In the cantaloupe outbreak, brush and felt rollers were contaminated with Listeria monocytogenes, and specifically in the 2014 caramel apple outbreak, after the environmental monitoring testing they did in the apple packing house in California, that was the supplier for the caramel apples, found Listeria monocytogenes strains on wooden bins, polishing brushes, and conveyor belts that were similar to the patient clinical isolates. So this was an evidence that there was a cross-contamination between those equipment surfaces and the apples. In addition, there have been so many recalls associated with packing equipment surfaces contaminated with Listeria monocytogenes related to fresh produce. Like in 2014, they recalled peaches, nectarines, and plums. In 2018, green beans and butternut squash cubes. In 2018, fresh avocados. And in 2020, fresh lemons, lime, oranges, and apple slices, mangoes, and pineapples. So we know the importance of having a good sanitation and packing equipment surfaces to prevent those outbreaks and recalls. So to do this research, we wanted to focus on surfaces that were most prevalent to have Listeria. So we based this sanitation research on our previous research, where we evaluated food contact surfaces that had the greatest prevalence of Listeria in apple packing houses. What we found was that polishing brushes and their fans, and dryer rollers were the sites with the most prevalence of Listeria. So with this science-based data, as Alexis said, you can focus your environmental monitoring programs and sanitation programs to see where you can focus your efforts for your sanitation crews. So as I said, this objective was to evaluate the efficacy of different combinations of cleaning and sanitation methods against Listeria on produce packing equipment surfaces that have been found to have a higher prevalence of Listeria harborage. So we evaluated different sanitizers and cleaners. For example, we evaluated PAA, chlorine, and alkaline detergent, the Critzer cold wax strip plus, and the use of steam. We selected these food contact surfaces, as I said, that were the most prevalent for Listeria species. We evaluated two material types of the polishing brushes, 100% nylon and a horsehair mix. Those are the most commonly used materials in the fresh produce industry right now. We also evaluated these brush rollers made of 100% polyethylene that are typically used for the washing step. For dryer rollers, we also use two material types, stainless steel and Teflon wrap rollers. And also one of the sites that are very known for having not having a good hygienic design are those interlocking conveyor belts because of the joints and those holes that they have in between. So we also evaluated this surface. So we evaluated 7 treatments and a control treatment. And we, for the first 2 treatments, we applied the sanitizers only. We applied chlorine at 200 ppm and PA at 500 ppm. Those concentrations were based on the maximum allowable concentration by the EPA label for food contact surfaces. Treatment 3 and 4 were the use of a detergent plus the use of either sanitizer and doing a manual scrubbing. Treatment 5 was the use of steam, and treatment 6 and 7 was the use of a degreaser plus the sanitizers. As you can see, we applied all these treatments to all the type of surfaces and materials, with the exception of the brush rollers, because of the use of the degreaser and sanitizers were not applied in the brush rollers since they are typically located before the waxing, so they don't get those wax residues, so they don't need a degreaser to be clean. So this is the methodology we followed. We inoculated our brushes that were clean with Listeria inoculum, and we applied the detergents, followed by scrubbing with another brush by hand. And then we did the water rinse and the application of sanitizer using this sprayer. We also applied the sanitizers only, and then we also applied the steam at 95 Celsius degrees for 15 seconds. This time, 15 seconds, was determined on replicable industry practices and also the heat resistance to damage of these bristles. After that, we enumerated Listeria inoculum and wax plates and determined the log reduction of Listeria. The methodology for the other surfaces that get this wax application in a common packing process was that we inoculated Listeria and then we applied the wax on the surface. For the polishing brushes, we applied a greater amount of wax since they get this wax through the wax nozzles directly. And for the interlocking belts and rollers, they do get the wax, but they get wax carryover from the wax produce. So we applied a less amount of wax for those last 2 surfaces. And then, similar to the previous slide, we applied the detergent plus the sanitizers, with the difference that we applied here a degreaser. So we follow the manufacturer instructions for the use of this degreaser, leaving the surface for 15 minutes soaking with the degreaser and then rinsing with warm water. And then we applied the sanitizers. As well as we did the treatments with the application of sanitizers only and the application of steam. For this, when wax is applied, for example, steam is thought to be an all-in-one approach, as you can use it for cleaning to melt these wax residues and then use it as a sanitizer as well. So that's also why we use steam in this research. So these are the results that we got. And as you can see in the y-axis, you can see the log reduction. So the higher the log reduction, the more effective the treatment was. You can see here in the x-axis the different treatments by type of surface. So these are the results for the polishing brushes, the different types of rollers, belts, and brushes made of 100% polyethylene. We based our efficacy of the results based on a 5 log reduction, which is a recommendation by the EPA. We got a greater than 5 log reduction for the rollers, interlocking belts, and brushes. And for the polishing brushes, we didn't get a greater than 5 log reduction. And horsehair mixed polishing brushes were the type of surface that showed the least Listeria inoculum log reduction. But what do I mean by a 5-log reduction? And this is the reference that the Environmental Protection Agency recommends, and they require to demonstrate a 5-log reduction for target organisms on food contact surfaces within 30 seconds. And this is a 99.99% reduction of bacteria. So that's how we focus our results. And you can see those recommendations from the EPA and on this website. So specifically for the polishing brushes, which are brushes that are commonly used in the fresh produce industry. As I said, similar to apples, cucumbers, peppers, oranges, they also go through a waxing operation. So for polishing brushes, we should focus our sanitation in having an effective wax removal from the brushes. As you can see, we got only a 3-log reduction, for example, in the horsehair mix brushes, when only a degreaser plus PA at 500 ppm was applied. And for the 100% nylon, we did get 5 treatments where we got a greater than a 3-log reduction. So based on that, we suggest that 100% brushes potentially offer a better hygienic design compared to the horsehair mix. Specifically in the apple industry, for example, the packers prefer to use the horsehair mix because they say that they polish better the apples. But in the sense of a hygienic design, the 100% nylon could potentially have could be more effective. And then also the horsehair mix was harder to clean and sanitize due to the more staff configuration of the bristles they have. These are the horsehair mix polishing brushes in this picture. So you can see that the wax, the build up of wax is a lot, and they have a dense configuration. And still, a 3-log reduction means a 99.9% reduction of bacteria, as you can see in the table. Then for the surfaces that don't get a direct wax application, as those rollers and belts, that they get the wax residues, but through the carryover of the wax produce, we applied wax, but in a very low amount, and the wax residues were not visible. Like, you couldn't tell that we applied wax when we were in the lab doing the research. So maybe you would think they are clean or they don't have wax residues, but the wax was not visible. So for these surfaces, we got a greater than 5-log reduction when we applied either the detergent or degreaser plus the sanitizers. And as I said, when we applied the sanitizers, only thinking that they were clean, we, for example, got for the stainless steel rollers lower than 2-log reduction. So for those surfaces, it's important, very important, to have an effective wax removal before sanitizing to get this greater than 5-log reduction of Listeria. And then the results for the steam are in this graph. For all the surfaces, we didn't get a greater than 5-log reduction. Specifically for polishing brushes, we got a lower than 3-log reduction, and even for the horsehair mix, we got a lower than 2-log reduction. And also we measured the temperature of the surface when we applied the steam, and we didn't get a high temperature, first of all, because of the time that we applied the steam, and also because of the wax layer that was there and that didn't allow to get this heat penetration to the surface. So the main takeaways with this research that I wanted you to know today is just a reminder that first you need to clean or wash and then have an effective water rinse. Specifically in those brushes, when I was doing this research in the lab, they absorb a great amount of water and also of the cleaners or detergents, so they are very hard to rinse. And so then you need to apply the sanitizer, but once you have your surface that is clean and without a detergent, to increase your sanitizer efficacy. Also, to have an effective wax removal before applying any sanitizer, even in surfaces that do not get a direct wax application. Also, that 100% nylon polishing brushes have a better hygienic design than horsehair mix, and that steam applied on polishing brushes full with wax was not effective against Listeria. Future work that we are also recommending related to this sanitation part is to find effective dry cleaning methods to remove wax residues on surfaces that belong to dry areas, and to refine the application of steam to improve the heat penetration and its use in combination with another sanitizers or cleaners. Finally, I would like to acknowledge the Washington Critzer crew who helped me do this research, to the Washington Tree Fruit Research Commission for financially supporting this research, and to PACE International and Pandora Sales for providing samples. And with that, thank you. We will take any questions.
All right. Well, thank you so much, ladies. We are going to start our interactive Q&A. So for those of you who are with us and you want to submit a question, please submit in the Q&A chat box. And I will go ahead and start. Let's see here. All right, let's ask this one. Let's go to Faith. What is the right way to sample compressed air?
Oh, yeah. So I would start by saying typically on compressed air, we're also going to have a filter that's going to be around 0.1 to 0.3 microns that will, if there happens to be microorganisms in line, especially bacteria, it will help catch them. But as far as sampling compressed air, you can go a couple of different routes. Based upon the PSI that's coming out with that compressed air, you can do either indirect air sampling, right, where you have an impact sampler is going to pull air through at a certain rate and sample it for a set time period. That is typically going to be indicative of more indicator organisms rather than foodborne pathogens. And more commonly, you're going to find mold. And no, it may not just be the compressed air. You can also basically, if it is higher PSI, blow the air through essentially something to kind of muffle it. If we blew compressed air directly onto a petri dish at really high pressure, it comes out at such a velocity and the microorganisms can literally bounce off of it based upon their particle size. So we try and muffle it in some way if we're doing some direct compressed air sampling. But I would first start with making sure I had a filter in line, and don't just assume that because there's something in line, it's set up to catch microorganisms because there's vapor and all sorts of things, impurities we can try and remove through these filtration processes.
Wonderful. How about this one? How would you determine if positive results you got are transient or resident?
Oh, that is a great question. And so much we couldn't really cover based upon the time frame. So with the transient, I'm going to get a positive when I go through and do my sanitation event. Before I even get results back, right? I should be able to clear that positive. And through my vector swabbing, I would be able to acknowledge that, right? And so I would say that that's essentially the evidence is going to be in that I don't find repeat positives. Whereas something that is set up, and within your system and going to be a bigger issue to try and contend with is going to give me potentially repeat positives. And many times, especially in produce packing where we get so much higher pressure transient positives, I would say that a lot of the guidance documents out there are written more so for manufactured foods where we have kill steps. So know that some of that's written in a slightly different context. And I would definitely say that give a big shout out to the International Fresh Produce Association, formerly United Fresh. They have really great guidance documents that I really like and would highly recommend for those people getting started. It also walk you through that.
Wonderful. And that's a great resource that we will make sure that we make available to all of you. Here's another really great question. Why do you not go directly to the species monocytogenes if the rest of the other species in the genus are not problematic? Price? ?
Yeah. Well, typically it's a bit of a hot potato, and I'll give my perspective and then Alexis and Blanca can jump in. I would say that especially when we're trying to monitor for a food contact surface, we're never going to routinely sample for pathogens, right? Unless there's some really special investigation that we're going on and we don't have packing going on, right? So that's the main thing. We also have found that, we do have these potential issues that can arise that a lot of our indicators are not going to pick up on if we're only relying upon aerobic plate counts or coliforms tied back to Listeria monocytogenes contamination, as Alexis, kind of showed so well. So I think that's really when we come into the sweet spot of sampling for Listeria species, if we're concerned about Listeria monocytogenes. And then a lot of people, yes, price is a continging factor, right? So a lot of people continue to sample for Listeria species, even once we get past the zone 1 surface, and then just treat it as if it is monocytogenes and really hope that it's not, right? And you're taking care of it either way.
Yeah, it's always better to be on the safe side, right? Let's see here. Would it be necessary or would it be a necessary procedure to recall product that was processed during zone 1 testing of food contact surfaces for L species and it came back positive?
Yep. Our best regulatory insight comes from the FDA and their guidance document, which is very explicit and states that, finding Listeria species on a zone 1 contact surface does not necessitate triggering a recall, right, for monocytogenes contamination. They are very metered, though, in that you can't just keep on finding Listeria species and surfaces over and over again without proper corrective action. So that's why we really reinforce that, making sure that you're clearing these transient positives that will pop up every now and again so that you know that they're not contributing to cross-contamination of product.
Perfect. Just a clarification for you. On one of your slides you presented, Faith, you talked about that you didn't recommend composite sampling. They asked, what about pooling samples? Synonymous or?
I consider those to be one and the same. So the thought process is never for vector, like you will never want to pool when you're trying to sort out exactly how widespread contamination may be. It doesn't help you, right? As a routine, kind of approach, if you do pool or composite, so you're taking either multiple swabs and pooling them together or taking the same swab and sampling multiple surfaces, you really have to be willing to then kind of go back and try and untangle exactly where my positive is. And you have to be fine with that. Some people say I can stretch my sampling budget much farther if I routinely either pool or composite sample, whereas I wouldn't be able to sample that many surfaces. So I understand there's a trade-off. You just have to be willing to say, I don't know which of these 5 sites are positive, right? Once you get a hit, and be okay with trying to maybe potentially do far more vector swabbing in that case.
Perfect. So our next question, I'm going to kind of try and translate a little bit, but they wanted to know about how you would approach a follow-up swabbing, or would you approach it differently if you were looking to find the source in addition to finding out how widespread it went?
So essentially, like, how far out would I go in my vector swabbing?
Yeah.
I would say that your vector swabs, your first round, I tend to keep it fairly close to home, right? And I'm looking at both where the products, if I get in on a zone 1, I'm looking at where the product's going to, as well as where it came from, and then immediately radiating out to my other zones directly around that positive. Based upon what you find from that vector swabbing, then it could almost explode on you, right? Because depending upon how widespread these positives are, you may then really determine that you have a far more widespread issue. Hopefully not. Hopefully you're going to verify that it was a very localized issue and that your program, cleaning and sanitation program, was able to handle it.
Perfect. This one comes from Hannah asking about, can you explain in depth the benefits or reasons to test APC APC versus TC? So Alexis, do you want to take a stab at that? Because you kind of talked about all of that.
Yeah, I can take a stab at that. And I think I'm going to give an answer that I think a lot of us do, which is it depends on the kind of information that you're looking for. And it depends on how many of these organisms you actually expect to be there. So one of the things that we mentioned in that second presentation was that if these organisms aren't there in kind of higher numbers, it can be hard to tell. It can be hard to get really numbers back when you collect your samples to give any sort of indication of whether you're hitting your threshold. But something as well to think about is your aerobic plate count, right? This is probably our least selective method for trying to figure out what is present. So if you're using this for a sanitary quality piece, that's going to be a really broad, really pretty big group. And then your total coliforms is usually a little bit of a smaller group. So I would say my recommendation typically is see where you're at with what your loads are for those two organism types to see if you've got enough of them present to be able to actually use that information and have it be informative for you. But I, of course, want to ask if Blanca or Faith also have any other suggestions that they would include in this discussion as well.
Always expect your coliform levels are going to be much lower, as Alexis mentioned. So your dynamic range is going to be far more limited. So sometimes that's why people like to look at their aerobic plate counts, because they're, our biggest net, right, that we can cast as far as bacteria go. And before and after sampling, if we go that route to try and establish efficacy of cleaning and sanitation, we get far greater dynamic range within that.
Wonderful answers. Exactly what I was kind of thinking. Let's go. So Blanca, you've got a couple of follow-ups to your really neat research that you presented. Were other sanitizers evaluated during this experiment? I noticed soap, degreaser, and PAA. Did we evaluate quat too and its effectiveness?
So we did evaluate two sanitizers, PA and chlorine. We did not evaluate quat, and we would love to.
I like that. Sounds like a really good future research endeavor. For steam treatment, does the duration therefore have to be increased above 15 seconds up to a minute or so, higher steam temp?
So we applied steam that also was like with pressure. So, and we'll, like I did a very small piece of brush and 15 seconds was a lot. And longer time start to damage the results.
Mm-hmm.
So we couldn't recommend a longer time. And also, it's not a replicable practice that you can see in an actual industry setting.
Wonderful. This is kind of a fun question. What is the relationship between having desirable microbes in or on food and controlling undesirable microbes? And I'm going to throw it out there to food and food contact surfaces. Who wants to take a stab at that one?
I would say so many times we are really focusing on really either trying to get at, do we have a potential pathogen cross-contamination issue that we're not even, we're honing in far greater, right, than we would for our beneficial microflora. There's a ton of work that's been gone on, that has gone on, kind of showing that generally speaking, our foodborne pathogens are fairly puny whenever they get some competition. And so, yeah, I think that it's interesting research. I don't know that I really have an opinion on it as far as environmental monitoring goes. A lot of times we're not going to catch that beneficial microflora, though, based upon the targets we're actually looking for in an EMP program.
Thank you. Let's do this one. Can you talk about the pros and cons of developing an EMP for small and diverse farms with packing areas that might be semi-open to the environment and that do not have a dedicated food safety manager?
Yep. I can take that one. I think that it is nice to get started. And I would start first with trying to work on how good my cleaning and sanitation practices are, rather than focusing on foodborne pathogens specifically. because we find that the better our cleaning and sanitation programs work, the less likelihood we should have with food pathogen specific issues. So I think that is a size and scale connected or relatedness. I would say, when it comes to diverse, smaller farms, looking at beyond just trying to sample from our indicator organisms after cleaning and sanitation event, also involving ATP to help inform our recleans when they're necessary, and something that can be done for after you invest in the reader, roughly $1.50 a swab for doing that ATP sampling. With the knowledge that it's just telling you how well you've cleaned, right? It's not really linked to any microbial population. I would also say that, really thinking through things we talk about more manufactured foods, order of the crop that you pack out in that packing house, right? We're going to want to start with what should be our cleanest crop coming in. And, if we have root crops and we have some leafy greens and we have some tomatoes, well, then I'm going to definitely process that root crop last, right? And along my shared surfaces. But I would just say that I would focus first on trying to make sure I had all those prerequisite programs really buttoned up before I started throwing any resources at an environmental monitoring program at all.
Yeah, and I think that makes a lot of sense with some of the things that we've seen out of the investigations about how important cleaning is and being able to verify that. Let's do this one. If a positive Listeria is found on a food contact surface in routine EMP work, should a company then request confirmation testing to see if they have Listeria monocytogenes?
I would say that's going to be very specific to the company themselves, right? I think that if you get a Listeria species hit on a zone 2 surface, you have to really look at what does it change your approach any at all, right? We know that in some past research, Laura, I believe that you've presented on it or been a part of, roughly half the time it's monocytogenes, right? So in some instances, up to half the time it's mono. So I think it really depends upon, the structure and organization. And a lot of packing facilities that I've worked with in the past have not necessarily wanted to go there, right? They wanted to keep it at the species level and then respond really robustly. And, at the end of the day, I think as long as you clear everything, then I think you're doing your job, right?
Yeah, and I think I would just add on to this, only because I dabble in this particular topic, is that, time sometimes is not your friend. And so if you wait and you request confirmation, that is slowing you out of the gate, right? And so sometimes I think it's better to have, your program in place. And if you get that L species hit, you've got your corrective actions ready and you're ready to have a robust response to it versus waiting, because ultimately, will that or won't that change, right? We don't really want any Listeria in our facility. So anyway, all right, let's do this one. This one says, this is a question for Faith. Can you help me to understand why we should stop production if we sample zone 2 area for pathogens?
Yep. So it really comes down to looking at how likely is that zone 2 going to interact with our zone 1s, right? And many times, if we showed everybody the same picture, right, it's like, oh, is this crossbar a zone 1 or a zone 2? That is really what you have to think through when you get back. And ultimately, you're going to find these hits when you're in the middle of production in a day. And so it's all dependent upon, based upon your analysis, how likely is that zone 2 service to influence a zone 1. So that is why, as something to think through, you should have in place, am I going to stop and am I going to reclean right away, right? Because I'm very risk adverse though. And I'm also spending other people's money when I'm up here talking. But as an organization, if you go through and you get a salmonella hit or Listeria hit on a zone 2 surface, you're in the middle of production for a day. Do you want to keep on producing with that knowledge, or do you want to stop, try and, even though it's downtime, bring in a sanitation off schedule, do you want to have that be kind of your leading approach?
Wonderful. What percentage of samples can I allocate to food contact surfaces versus non-food contact surfaces in a program?
I would say well, it depends on what you're sampling for. If it's something like Listeria species, I think it's really beneficial to have some food contact surfaces in there. Some people have been very averse to sampling any food contact surfaces. The adjacent areas, as Alexis mentioned, you can get ideas, but I think knowing exactly what's on that surface, you may be surprised when you actually start sampling the surfaces themselves. It's all rule of thumb, but I would say about a quarter, from food contact surfaces, and at least if I was sampling. Again, if we're talking about cleaning and sanitation verification, then I would sample far more food contact surfaces, right, for those surfaces. I don't know. Laura, you're an expert, and Alexis and Blanca, I'm sure you all have opinions as well.
Well, I was going to say, Alexis, you've done a lot of research. What do you think?
Yeah, well, I love that at the very end you pulled in that. Yeah, I think it depends on what you're sampling for and why. And so I completely agree that if it's a cleaning and a sanitation purpose, I'm going to prioritize my food contact surfaces way more than I am some of those non-food contact surfaces, just because how am I using that data, right? I'm using it to let me know if I need to re-clean, re-sanitize. And so all of that being from the perspective of making sure that our food contact surfaces are safe for food production. So completely agree.
Yeah.
Wonderful.
The one thing I will say is zone 4s, like I put them in the least category, right? And as far as
Yeah.
but,
Absolutely. Yeah, absolutely minimal. And I think you're right. When you're doing the Listeria species and stuff, yeah, I've got majority of them are on the non-food contact surfaces because they're my early warning detection, right? I don't want to get them on my food contact surface. If a vector swab comes back positive, at what point do you stop vectoring? Because when do you quit vectoring, right? Until it finds it?
General rule of thumb is you want 3 to clear any positive site with 3 follow-up negatives.
Perfect. This is a good one for, I guess, maybe some of our recent graduates. How long does it take for Listeria to grow on food and food surfaces?
It depends on so many factors. Nutrients, your cleaning and sanitation,
Temperature,
and pH. So many factors. So
absolutely. I love that. You're getting ready to do my job then. It depends. And it's called, what is I guess we've got, about 3 minutes left here. So let's see if we can power through these last couple. What is the impact of the containers used for harvest regarding the entrance of Listeria to a packing house? So let's talk a little bit about containers.
Yep. I think that, when we're thinking about binning out and other, containers moved by hand, really look at, we know Listeria is soil borne. Let's try and restrict, direct soil contact. So having pallets where we're staging things for our bins as they're, moved out to the field, anything we can to prevent that direct soil contact, especially considering most of this, especially if we're thinking about bins and even handheld containers, they're staged on top of one another. So what's on the bottom of something is going to now reside on top. I also think it's really important for us to think about how recently some of these bins have been cleaned and sanitized, right? And especially because that's scrutinized more and more under the produce safety rules. So again, I think half the barrier here is making sure your harvest crews know that it needs to be a clean container they're packing into. And if it's not, don't use it. So I think that's one of the easier things because it's not invisible like most of the things we deal with when it comes to microorganisms.
Wonderful. We had a question about how do you recommend handling vector swabbing in zone 2 versus zone 3?
Oh, really great. So if it's zone 2, I'm going to have far more kind of leaching over into the essentially the food contact areas, right? And also looking at what may come in contact. With zone 3s, they're kind of unwieldy. I mean, some things are in fixed place, but other things that fall in that zone 3 are going to be like carts and forklifts and all these other things that are kind of adjacent. So really thinking about where all does that go, right? And what exactly may I need to look at as far as flow of that product that sample or that positive sample came into. So I would say essentially with zone 2, if you're going to try and vector out onto food contact surfaces, I would really look at that's the primary difference. And with our zone 3s, they're kind of our early warning stage. So just trying to make sure it hasn't moved on to zone 2. This is where I'm going to focus on there.
Wonderful. Well, thank you guys all so much. I want to thank our speakers today and our audience for their fantastic questions. These were some stellar, awesome questions. I thoroughly enjoyed them. Also, as an update, several individuals have requested we develop a YouTube site or page for webinar recordings, our key takeaway documents. So please stay tuned. We will work on that. We've also got a couple of requests to try and translate some of our resources into Spanish. We will also try and do that as well. We look forward to seeing you next month at our webinar about supply chain integrity on Thursday, May 12th, 2022. If you hold your phone's camera up to the QR code on the screen, it will take you to the site where you can register for this event. As well as all of you who have registered, I will send you a reminder email. So thank you again, and have a great rest of your week and a happy Easter.